a good morning to everyone. So let me thank once again to Professor Roberto Tobrosa, for whom I know for more than 20 years now, for this invitation. You need the speaker. Oh, okay, I can put this one here. Yeah, that's also a good idea. Yeah, so thank you once again, Roberto, and great to see many friends here, including Marco, Silvio, Luigi, and today I met several other colleagues like Marco and many other colleagues that who are not from Bologna, but they have come from other places to join this seminar. And Bologna is very nice place, and this is my third visit. I always love to come here. So in this presentation, I will try to tell little bit about some genomics work what we have been doing. I know that in announcement probably the title was Breeding 5.0, which Roberto and Marco they put from somewhere. I don't know from where. But then I told, well, I work on genomics, so please excuse me. I will talk about genomics. Now, many of you may be aware we are having lots of challenges, especially in terms of that feeding the world. And why we need to think about that? One is that the world will have overpopulation growing more and more. So we need to feed this ever growing population. Second, in the current scenario, we got lots of geopolitical events and they are also affecting the food supply across the world. And of course, we are aware about climate change. The world is becoming warmer, though there are different predictions, but you take any prediction projection, this is very clear. The temperature is increasing, which will be adversely impacting crop productivity, mainly in the developing world. And also, we got the issue about the malnutrition, that even in some countries, people can have enough calories, but then they are having the deficiency of proteins and many other things. So there are lots of challenges. So I think that for addressing these issues, it's very important that we need to have the diversification of agri-food systems. So not just one crop like wheat or barley or durum wheat or bread wheat, but not only the pulses, fruits, vegetables. So I think these are important. We need to have the holistic view. And this is very good that from University of Bologna, Roberto and his colleagues, they have done amazing work in this plant genetics and breeding, which has impacted the global science. And I will also mention in few other slides later. So this is some of those issues we need to do. Now the thing is, are the challenges are new? Probably not. Even way back in 1960s and 70s, and I'm talking that many of us young people, we were not born at that time point, but I'm originally coming from India, so I have always heard the stories from my parents, grandparents, that in 60s we had that very scarcity of the food in India, South Asia, Pakistan, Bangladesh, and many other countries. So to address this issue, when we had these problems, after that, under the leadership of Norman Borlaug, we had this Green Revolution. And for the Green Revolution, there were many other heroes, including M.S. Swaminathan, Gurdev Ghosh, John Longping. And these gentlemen, they did amazing work, especially in the area of cereals. Many of you may be aware that how they introduced dwarfing genes. They changed the plant type. They enhanced the crop productivity. Country like India, which was just producing around 40 million ton wheat way back in 1950s, now they produce more than 110 million ton wheats, and the country has become a major exporter of wheat. So this is the power of the technology, power of the genetics, agronomy, etc. So these were that. Now, not only in India, if you see around the world, impact of the Green Revolution age, the cereal production from 60s to now has increased 3.5 fold. Yield has increased 3.1 fold. On the other hand, we are always talking about the population growing 2.6 fold. So basically the food production has increased more than what population is growing. And land use, you can see at the bottom, is not increasing for the cultivation. So this is the some of the positive side. Green Revolution also established a lot of infrastructures in many countries that now people have the possibility to have irrigation facilities, mechanization, but at the same time they also had liberal use of chemical fertilizers. So this is sometimes negative side. But cereal in yield, as we said, that average or so, so this has been increased quite full, quite high. This is the world map that the current stage, when you see these low yield in the brown color, that higher yields in that green color, if you see around the world, go to North America or in Europe, Western Europe, you see lots of green color. Go to India, you also have that thing, but majority of places in Africa, we don't have the higher yield. Maybe one of the reason is that these countries, we did not use much fertilizers, we don't have much technology, so these are the areas that we need to work. I'm not telling that we need to use the heavy use of the fertilizer, not at all. So what we need to do now in the current scenario, we need to see the way that how we can have our agri-food system sustainable, so that we should not be thinking that when temperature is going higher, we need to produce more. Carbon dioxide should not be going in the greenhouse gases, which need to control. We don't need to have the more chemicals in the soil. So there are a lot of things, and many things can work on those aspects. Now going back and this connections that food security, and I always tell in the different platform, not that because I'm here, and some of you, if you have not heard, I was asked once to give a TED talk, and in my TEDx talk, I also talked about Bologna. Why? That this was 2003, as Roberto mentioned, that we had this 50th anniversary of the DNA. At that time I was young, and Roberto was also very young, with black hair. Now he doesn't have the black hair, all the hairs are gray. So this is the hair of the wisdom. But anyway, I was sitting somewhere in that corner because this Congress was in the same hall. And in this Congress, even today we remember that all the topmost people working in genomics, breeding, agronomy, all of them from America, North America, from Africa, Asia, Europe, they were here. And I was really very lucky one that I attended this conference. I met several people, and that now in the center, if you see this picture, Norman Borlaug, Swaminathan, and Gurdev Ghosh, who were the main people for that Green Revolution, they were here. I had a pleasure to have a picture clicked with Professor M.S. Swaminathan. At that time point, I was working here in Germany. I came with my boss, Professor Andreas Graner, to this place. And I remember that Norman Borlaug, in this hall, he challenged the next generation of scientists, and he was talking that now we are talking about the gene revolution. I would like to challenge the next generation of scientists to embrace those new technology and to address the real world problems. So this was good, and I will connect this thing later. So in this 2003, then I went back to my institute, IPK Gattersleven. I started to have the discussion with Andreas Graner and some other colleagues, and I will come back to this in a minute. But that was the 2003 where I had this, I came to Bologna. So thank you very much, Roberto, for bringing me to this meeting. And then at later stage, I came in 2010 in Bologna. And when Roberto and Marco, Silvio, and many other colleagues, they organized this meeting called Genomics of Plant Genetic Resources. And the good thing is that whenever I come to this place, I always get the new connections, new network or so. And as a result, as Roberto was saying, we started to work together. We published 2 books, Genomics Assisted Crop Improvement from Springer. Then another set of the book, we had the Translational Genomics for Crop Breeding. So a lot of stuff. During last and after moving from Germany, I was working in ICRISAT, one of those global CGR center, worked there for about 17 years. But when I was in India, we have been working together on different aspects. Roberto, you might have come to India maybe more than 5 times and different meetings. We remain in touch, and we are working in close collaboration now with Marco on many other aspects. But anyway, so what I want to mention that then, this is also very interesting thing that whatever Congress Roberto has organized, so he has infected me with this organization of the Congress. So for instance, when he organized Interdrought, I also organized Interdrought. In India, when he organized the GPGR, I was also asked to organize the GPGR. So I was thinking that why this is happening always this one way around, that like Roberto organized first, then I organized later. I said we need to change it. So what we did, that this was by chance. Last year, we organized the International Wheat Congress. So, Roberto, this was first time that I organized the Congress first. And then I passed this baton to Roberto. And he will be organizing this International Wheat Congress next year in 2026. So this is one of the reasons that I'm here to interacting this different colleagues, that how will be the Congress. Anyway, coming back to this 2003 meeting in Bologna when I came here. Then in 2005, we had rice genome sequenced, 2005. And this Congress happened before the genome sequencing was happening. So in 2003, the first human genome was decoded. At that time, rice, Roberto at that time, Steve Goff was also here, that who sequenced the rice genome, the thing. So Roberto brought a lot of people. But anyway, so with Andreas and also another collaborator, Mark Sorrells from Cornell University, we were talking, and I was always saying that now when a lot of genomics, genome sequencing is happening, how genomics will be used in the breeding program. So we provided a concept or framework, we call it genomics assisted breeding. In 2005, in the Strains in Plant Science journal, in the 10th anniversary issue, Feeding the World Plant Biotechnology Milestone. And this paper has become a landmark paper. Now you go to the different conferences or book chapters, books, that they always have a section called genomics assisted breeding. In this paper, we provided the concept that how the genome sequencing, modern approaches of the breeding, etc., will be used. So after this 2005, with this inspiration from many big people, including Norman Borlaug, Mike Gale, and Roberto, or many other colleagues, Jeff Vanitgen, so I met several people, with an objective to contribute towards the global food security. I joined International Crop Research Institute for Semi-Arid Tropics, which is one of the CGR research institute based in India, but we were working there in Africa and many countries, 47 countries. So we were working on those crops, chickpea, pigeon pea, or peanut, which were not very important crops for the Western world. We did not have much genomic resources. And I wanted to develop genomics assisted breeding framework in these crops. One of the key thing is that you need to have the genomic resources. So what we did, I started to develop some international consortium and started to use my knowledge, what I generated or what I gained in IPK Garters level, and I had from my different collaborators around the world. And we started to decode the genomes way back, and we started this work in 2012, pigeon pea, chickpea, pearl millet, in the case of peanut for many crops, and of course sesame, mung bean, ajoki bean, etc. And we feel very privileged or proud or very happy that our lab has decoded genomes for more than 12 different crops, more than a dozen crops. So this is really good that they have provided some genomic infrastructure. At that time point, we were using Illumina sequencing technology, which was not very big or very hi-fi technology. So genome quality was not that great. But what we did, we kept on evolving with the technology. We kept on improving those genomes. And we reached to the place that when we can develop the gapless genome assemblies, and this is that example in collaboration with Henry Wayne, where we have done this work, and this in the soybean, that you can identify the telomere, you can have the gapless genome. And now we are also moving towards developing telomere to telomere genome assemblies. So now this good news is that we got lots of technological advances. So you can really develop high quality genome assemblies. And we have started to use this technology at Murdoch University now in Australia, in wheat, in legume crops, and in many horticultural crops. Horticulture crops, and I will discuss later, including the crops like banana, custard apple, passion fruits, pineapple, papaya, etc. You can see these pictures. So we have been using these things in the different genomes. Now, if you would like to improve these things further, we know that during the course of domestication and breeding, we have lost several important genes in the breeding germplasm. So now this is the time that we need to understand the genomes of the wild species. People were talking pangenome. We thought that pangenome is always to assemble those gene repertoire of a crop species with the objective to introgress the genetic diversity from the wild species. We provided a concept of super pangenome, in 2018 to 2022. And in the superpen genome, we suggested that we need to develop the genomes of the different species of the same genus. And at that time, though, we suggested that from each species, we should have at least 10 exosomes, and then we should combine these things together. We should come with the superpen genome. But nowadays, if you see the papers, there are lots of paper coming in the area of superpen genome. And with this objective to catalog the genetic diversity, when I was at ICRISAT, we sequenced around 292 genotypes of pigeon pea coming from all the different places of the world, around 429 lines in chickpea, did a lot of phenotyping, and did that identified the loci associated with the different agronomic traits. I will not go in the details, but I just would like to highlight some key features. Then we went one step further. In the case of chickpea, we sequenced more than 3,000 chickpea lines at the whole genome level, and then we developed the chickpea pen genome. And this pen genome suggested that if you take only one genome, you may miss many genes of that particular species. For instance, by sequencing 3,000 genomes, we found that 1,582 new genes were found. And this pen genome we also used to identify the haplotypes for yield-related traits. And we provided a concept that how the pen genomes can be used to catalog the haplotypes, and these haplotypes can be used in the breeding program. One of the key things of my research has been not just sequencing the genome, but utilize them in the crop improvement program. And we have been doing this kind of work, for instance, in the case of pea, in the case of azuki, where we are sequencing large number of exosomes. Recently, in the case of chickpea, where we are having 8 different wild species, and what we did that we developed the genome assemblies for all these different species. We put this thing together and we developed the Sicer superpen genome. And the Sicer superpen genome has provided several interesting genes which are not present in the cultivated one, and they can be used. We went one step further and we have sequenced more than 10,225 chickpea lines. Probably this is the largest plant genome sequencing project so far, where we have sequenced more than 10,000 lines. So analysis is still underway. But here, what we have done that we have been doing really very interesting thing that when you do 3,000, what you miss, which you would, for instance, many people ask, what is the fun to have this 10,000 now? My plan when I was at ICRISAT to sequence entire germplasm collection, we had only 20,000 exosomes. This could not be fulfilled because I moved to Australia. But anyway, now the thing is that based on these different analysis, we see lots of haplotypes, which is having huge potential to improve the chickpea crop or so. Now, when we are in Australia also chickpea is very important crop. We were not having those kind of genomics advances earlier. So what we did, we targeted around 15 elite chickpea varieties, and we developed the chickpea pen genome for that Australian lines. And now here you can see these are the different kind of desi and Kavli chickpea. They are that whole genomes. But this pen genome, you can see the different variation. I will not go in the details. If somebody is interested, will be happy to see. But one thing is some important genes which are associated with the yield under drought conditions, because these genes, they came from one of the QTL hotspot. Roberto, Silvio, they have done a lot of work on the QTL cloning or so, and we also cloned one QTL for root traits in chickpea. So some of those genes coming from that root trait QTL, we analyzed, and we analysis of chickpea varieties of Australia indicated that they are missing some important superior haplotypes for drought-related traits which are present in the varieties released in India and Africa. So now this is the way forward that how we can introgress those haplotypes here, and we can do this thing. Now, when you work in Australia, this is impossible that you will not work on wheat, because wheat is one of the leading important crops in Australia. We work together with the Chinese group led by Shuyong Zhang from Chinese Academy of Agricultural Sciences, and we sequenced 17 cultivars. And then we have done this thing analysis, comprehensive analysis, and they have told a lot of important things, especially from the evolutionary insights perspective, that how the wheat has moved or transition from spring to winter type just because of some gene mutation in VRN-A1 genes and etc. And these are the important area for environmental changes. This prompted us to take up another study. And now we are working together with Martin Master and Manuel Spangel from Germany. And plan is to assemble a wheat pen genome with 100 wheat varieties. And we are very much pleased that from Italy, we have Roberto, Luigi, Marco, and many other colleagues. And you can see that in these pictures. So, Luigi, you were present in all the three meetings. Roberto was only in two. So I can always say that. And Marco, you were also in all three? Probably only two. So, Luigi, you are consistent one, which is good. So anyway, so what we are doing that we are having these different countries, and plan is to have from each country 5 to 10 wheat varieties, so that we can understand that adaptation for drought or heat or winter across the different crops. Plan is to have the high quality reference genome assemblies for about 100 wheat varieties, genome annotation for all, comprehensive wheat pen genome, and haplotype catalog for wheat. And all these resources will be available in public domain. We are work is in progress, but we finished around 12 wheat varieties, genome assembly and annotation. Now this completed about 47 different genome assemblies. And very quickly, what we did, we analyzed these Australian wheat varieties with China. And you can see that there are some changes, but very important thing is that genome of Australian wheat varieties, they show a relatively good collinear relationship, but there are structural variations in the genome of some individual Chinese wheats. And now we are having this Chinese spring, which is that's considered as a reference wheat genome. Genetic variations in the genomes of Australian wheats are higher than those of the Chinese cultivar, but Chinese wheat is having a better genetic diversity. I will not go in the detail. I had some slides, but okay, the slides are in the other presentation. Anyway, so those kind of work is moving ahead. And now another thing is that when you develop those genomic resources, how do you use them for the crop improvement? I will like to give some examples from our work, what I was doing at ICRISAT earlier and also in Murdoch University. So the genetic dissection of those different traits is very important. So for instance, now in Australia, we are working on nitrogen use efficiency. This project is funded by GRDC. We are the national leader in this project together with the University of Western Australia and AGT Wheat Breeding Company. And this AGT Wheat Breeding Company, we are working with them. They are doing that phenotypic evaluation in multi-locations trial around the whole country. We are doing a lot of genomics work, metabolomic analysis, and then we are trying to identify those genes, understand the molecular and biochemical mechanism, and also trying to use the CRISPR-Cas technology for the gene validation perspective. Another project in wheat is about the heat stress tolerance. Similar kind of a story, targeting about 345 wheat varieties. So phenotyping in Australia, we do a lot of phenotyping work with the wheat breeding companies and go at the multi-location field trials. And we do some work in the greenhouse as well. But again, the idea is the genome sequencing, haplotype analysis, and then target those kind of traits. Pulses, because I worked on pulses for about 17 years and I'm continuing this love for pulse crop now as well. So for even that now in Australia, we are leading this genetic gain initiative in chickpea, fava bean, and lentil. And what we are doing here, lots of partners. We are developing these diversity sets, characterizing them at the sequencing level. We are using AI-based approaches for haplotype stacking, and also bringing some diversity from ICARDA and then developing this pulse-based database. We are also working with another project together with the University of Queensland with Michael Udawardi, where we are interested to understand the nitrogen fixation, symbiotic nitrogen fixation, and lots of projects in horticulture crops. So for instance, we are developing the national platform for Australian horticulture, and here we are working on these 5 different crops: papaya, banana, pineapple, custard apple, and passion fruits. Another one is that we are using these kind of approaches to develop the cost-effective genotyping platform. And now recently, this is the WIC project, which is $41 million initiative led by Queensland University of Technology, but we are node for Western Australia. And we are working in Western Australia on 3 crops: mango, almond, and apple. So I mean that this is another thing that how we can use the genomics technology. So we are interested to identify those kind of things. Way back now, once you are having that traits identified, genetics understood, we would like to use them for the crop improvement. And here I will like to take you back again to ICRISAT work. So when I was at ICRISAT together with our colleagues for about more than 10 to 15 years, we identified number of markers associated with different traits: drought, heat, salinity, ascochyta, groundnut for many traits, and pigeon pea. This is great, published a lot of paper. But what we did, that we used this information and improved the crop varieties. So for instance, for drought, we had, in the case of chickpea, we identified a QTL hotspot which contains the QTLs for more than 20 different drought tolerance component traits. We did a lot of work, and then based on these lot of work, and we started this project sometimes in 2010. So your first paper came in 2014, and last paper we came in this 2022. So around 12 years work. So what we did during last 12 years, we have developed these population, high resolution mapping population, did a lot of map-based cloning, and eventually we identified or validated that one gene, C8TF4V gene, is associated with the drought tolerance. So this kind of thing in several crops we did the QTL mapping, fine mapping as well, but in some other thing we just use those QTLs work. When we had these QTL kind of thing, and we were working in India and Africa, and we asked our breeding program that how we can use this genomic technology in the breeding program, we found two issues. One, the genotyping cost was quite expensive. Second, many breeders, they were not trained in those technology. What we did, we were working with the Gates Foundation, and we established one platform called high throughput genotyping platform. What does it mean? That any breeder, when you are having these breeding population, they can do the genotyping of one line with up to 10 SNP markers, which are related to the foreground selection, only in $1. So $1 includes the DNA extraction cost and the 10 SNP genotyping. So this is the way that we made this genotyping cost effective. And in this project, I was leading this project. This was working with 8 different CG centers, 13 NARS partners across the 28 countries. So this made huge impact on deployment of genomic technology in the breeding program. Second is, and Roberto was also there in several programs. So we trained the breeders in genomics assisted breeding, and we were bringing the breeders from all around the world. But at that time point at ICRISAT, I was working more for India and Africa and South America. We trained more than 485 breeders from about large number of countries. So we put these things together: genomic technology, cost effective breeding programs trained. Then people started to use the genomic technology. So this was another thing that we were organizing lots of conferences, exposing them to the modern technology, inspiring this next generation of the breeders. And after that, we started to deploy those molecular technology, and these programs were led by the breeders. So for instance, here, this is an example. Several research institutes, several breeding institute from India, breeding and agronomy institute from India, they worked together. We provided the support to them that how you can introgress these QTLs in the elite varieties. And then in these countries, we are having multi-location field trials. As a result of those multi-location field trials, many varieties were released. They are grown by farmers, and these varieties are having more than 20 to 25% higher yield what breeders or what farmers were harvesting earlier. So now, for instance, this is the drought. Drought is a very complex trait even for drought, for the high yielding varieties coming from this work. Not only in India, in Ethiopia, Kenya, Tanzania, in several other countries, we deliver those varieties to the farmers. And now the same thing for the other diseases like fusarium wilt and ascochyta, etc. So chickpea alone, more than 15 to 20 varieties have been delivered through the genomics assisted breeding. And some of those FAO documents that we presented the genomic assisted breeding delivering superior chickpea varieties in India, Ethiopia, Kenya, and Tanzania. So this was work for several years with large number of partners. But the good news is that the crops where we did not have anything earlier, now we got the products through the genomics assisted breeding, and farmers are growing there. Same thing, we did the work in the case of peanut. So in the case of peanut, we had the foliar diseases, high oleic acid. We are having more than 10 different varieties released in India. Same thing in Africa, especially in Ghana, Nigeria, Burkina Faso, and many other countries. So those varieties, they have been delivered and farmers are growing them. Pigeon pea is in India, so there are lots of varieties. So this is in the developing world. Now we are deploying this kind of technology for the traits like nitrogen use efficiency in Australia, and we are moving ahead together with the AGT wheat breeding company. Same thing in the case of chickpea, together with chickpea breeding Australia. Only the difference in India and Australia is in Australia, breeding is being done by the breeding companies. In India, this was in India, Ethiopia, Kenya, Tanzania, they were done by Public Breeding Institute. So now we work here in that breeding companies, and now we know that these are the chickpea varieties, but then some of those QTL, they are missing in those varieties, and now we are trying to introgress them here in Australia. So this is about that Australian system. Now, very quickly, in next 3-4 minutes, I would also like to highlight one more thing. We can do a lot of work in the area of genomics. We can understand the genes, we can understand the genome, superpen genome, we can have a haplotype catalog. Okay, then I will speak slow. Thank you. So we can do a lot of work on those aspects. Publish the paper Nature or Nature Genetics. That's fantastic for next generation scientists. They develop their career. But most important thing from our perspective is that how we can deploy them in the crop improvement. And I gave this example in the case of chickpea or peanut, that the crops where we did not have the genetic maps. When I joined Agri-Food in 2005, we did not have the genetic maps in those crops. Like pigeon pea, 11 linkage groups. When I joined, there were just 10 SSR markers, only 10 SSR markers published in one paper, Molecular Ecology Notes. So even you cannot do this thing. But now the good news is with the power of the partnership, power of that collaboration, and the technological advances, we have demonstrated that you can develop those genomic resources, use them for understanding genetics of traits, and then develop the varieties. Now, another thing that even if you can develop the varieties, it doesn't matter these varieties are developed through traditional breeding or genomics assisted breeding. One more point is missing, and this is delivery system. What does it mean? So when you got these elite varieties, in many countries, especially when you work in Africa or Indian countries or South Asian countries, seed system is completely broken, unlike the Australia or Western world, where you are having lots of seed companies and they are connecting that farmers, they are growing the new set of the varieties. What happens based on my experience? So even you have these varieties, they are not reaching to the farmers. And even if this reaching to the farmers, they are not having the good agronomy packages. So they cannot utilize the appropriate agronomic packages. So what happens? So you can improve the seed. DNA is inside the seed. But if you would like to realize the full potential of genetics, you need to provide the good package outside the seed as well. So we need to bring these things together. So we need to have the better agronomy. Sometimes because of number of regions, you got a lot of post-harvest losses. So even farmers, even sometimes you got crop growth higher, bumper crop, but then they don't have the infrastructure to store those things. You cannot get that money. And we thought that we need to address a lot of issues of these kind of things. Second is that now how we can work, that how we need to connect these farmers to the market so that as soon as they harvest, these are going to the market. Can we talk about the value addition so that we can have this beneficiary, the farmers and also consumers. So this whole chain is required, rapid delivery system is required. To address this issue, what we did, we worked with the Gates Foundation, and this is considered one of the flagship project of the Gates Foundation. What we did, we called it Tropical Legumes Project. This ran in 3 different phases. It started from 2007 up to 2019. And I was the project coordinator for Tropical Legume 3 project. They were my project investigator and other people. And I worked with these 2 gentlemen from the Gates Foundation. This was led from ICRISAT. We had collaboration with CET and IITA. So total investment across this 12 years, this was 67 million US dollar. And we run this project in 13 different countries in Eastern and Southern Africa, Ethiopia, Uganda, Tanzania, Malawi, and Mozambique, Zimbabwe, in several countries in Western Africa, Western Central Africa, Mali, Senegal, Burkina Faso, Ghana, and Niger. And in India, several states and also Bangladesh. So total 15 countries, 13 countries in Africa, and then 2 countries in Asia. We targeted these 6 different crops: chickpea, cowpea, groundnut, common beans, soybean, and pigeon pea. So this is a huge project. And I was traveling a lot in Africa at that time point in the different countries. What we did in this project, 3 things. One, develop and released farmers preferred improved legume varieties, because so that we were involving farmers in terms of the release of the varieties as well, so that farmers can be interested to grow these varieties. Otherwise, many times as a scientist, you can develop the varieties. Farmers say, we don't need these varieties. So you need to have the demand driven variety. So we targeted this kind of thing, and we have delivered more than 266 varieties during this total time frame. Second, strengthening the breeding pipeline. In the breeding program, how you can deploy the genomics technology, what is missing. So very small thing, as I was giving the example that we say, you don't have the DNA lab, doesn't matter. You just cut the leaves, send this thing to this company. They will provide the data. You just use that one. So this was the another thing, that is strengthening the legume breeding pipeline. And this is the way that we have been working in different countries, different varieties, etc. So this is good. The other thing is seed delivery platform, because legume crops, they are the self-pollinated crops. Many companies were not involved, but Gates Foundation helped us to develop those platform so that we can help them, and we have this program help 1,872 seed producers were engaged, and their job was that when we give the new varieties, they can do the multiplication of the seeds and then deliver to the farmers. So this was a big thing. And because of all these things, during this 12 years time frame, 304 improved varieties were delivered, 397,000 tons were produced, which were grown in 4.4 million hectare area, produced about 4.9 million ton seeds, which was worth for about $2.6 billion with the cost investment of $67 million. And the different analysis, economic analysis, not from us, they provided the data set that 22 million farmers were benefited from this project. Now, similar kind of technology should continue to be used. Sustainable thing. We trained more than 100 next generation of the breeders in those countries. Whenever I go to Africa or so, even now, not all 100, but at least 20, 30 of those breeders, they are leading those breeding program now, which is also one of those key things. Some of the examples. So for instance, in the case of Ethiopia, if you see that's improved seed production, earlier this used to be 1,000, now this reached 3,000. Earlier in common bean, 2,500 reached 5,000. In Uganda, if you see that how many households, they reached this thing from 164 to 560,000. For instance, in the case of Nigeria, in the case of groundnut and cowpea, earlier we did not have much seeds produced. Now they have got a lot of things in Mali, how things are improved. So that's across the different countries. This project has really helped a lot. And now we have documented all these success stories as well as failures as well as challenges. All these different things in different special issues, in the different books, etc. That, well, how we were this thing. And Gates Foundation, they did independent analysis of all these projects thing from Washington, from the Evans School Policy Analysis and Research. This is the Economics and Social Science Department. And those people, they told that this is one of the key flagship project of the Gates Foundation across a large number of projects. And then they have suggested this TL generated $16 on investment of $1. So I think that this is one of the success story, that how that full portfolio of this had very high social returns relative to the BMGF's level of funding. So this is that all documented, and you can see in this Gates related stuff. And for that work, we were awarded ICRISAT as a 2021 Africa Food Prize as well. So I think that this is that some of that success stories, that how by using this genetics, breeding, agronomy, etc., you can put these things. But this took a lot of time, a lot of energy at that time point. In summary, I wanted to mention crop diversification is essential for sustainable agriculture and ensuring food and nutrition security. Genomics, pangenomics, haplotype analysis, they can identify precise genes, genetic selection tools for genomics assisted breeding. Now, I have not touched because we are still working. Some groups are already in advanced stages. How we can use the artificial intelligence and machine learning approaches, simulation approaches to accelerate trait mapping, identify optimal parents, predict agronomic performance. This is the area for future. We need to work in this side. Rapid delivery system is readily required in the developing countries. In developed world, this is not a problem because companies they are taking care of this thing. International, national, and local government agency support, networking is very important. That's what you guys are here. I have been traveling. You guys keep traveling here and there. So this that we keep on working on those different aspects. So I think that this is also very important working in collaboration. So thanks to all the different colleagues, but right now this is my team from Murdoch University, my research group, more than 25 people or so, lots of postdocs, brilliant postdocs, PhDs, graduate students or so, and lots of partners. Even I'm there now for last 3.5 years, but we already developed a good network in Australia. And that's what I consider as one of my key strengths, developing partnership, networking, and of course, right now, lots of partners from the different countries, different people. With these words, I would like to thank you very much for coming here. We'll be very happy to answer any question for you. Thank you.